Recording in progress.
Now let's start with Prophase 1, where we are going to discuss about all the substages of Prophase 1, that is Leptotene, Zygotene, Pachytene, Diplotine and Diakinesis. So one by one we will discuss all these subphases. So let's start with first one, that is Leptotene. Leptotene, or we can say leptonema, which comes from the two words, that is lepto and nema. Lepto meaning thin and nema thread. Okay, so during this phase, the chromatin network, they condense and resolve into long and thin thread-like structure, that is what called as chromosomes. So let's see the diagram. Here is our cell. Inside the structure, that is nucleus. Here is our centriole. This is nuclear membrane. And here is nucleolus. Yes, here we can see our long and thin thread-like chromosome. So in this way, okay. So this is long and thread-like chromosome that is present where in this leptotene phase inside the nucleus. So each chromosome, you can see it consists of two chromatids but appears like single thread. Okay, leptotene. So here we can say beaded chromosome. Beaded chromosome. So here you can see beaded chromosomes and nuclear membrane and nucleolus are present. Now let's move on. Zygotene stage. Second is zygotene. So in the zygotene stage, you're pairing of homologous chromosomes. So let's see the diagram. Here's the cell. And yes. Here in zygotene stage, the homologous chromosomes, they will start make pair. तो वहाँ पे जो थ्रेड लाइक क्रोमोसोम्स थे वो यहाँ पर क्या करने वाले हैं, पेयरिंग फॉर्म करेंगे. सो लेट्स सी. Here is my first chromosome. Yes. Here you can see they start making the pair. As in the leptotene stage, here you can see thread-like chromosome. Thin thread-like chromosome. Whereas in zygotene stage, here we can see what? Pairing of homologous chromosomes. Pairing of homologous chromosomes. Okay, this is the cell, this is the nuclear membrane. So here you can say nuclear membrane. Here is my nucleolus. Okay, nucleolus. And here chromosomes. Yes. So here pairing of homologous chromosomes takes place. And homologous chromosomes are seen as bivalent. Very important part. Homologous chromosomes are seen as bivalents. And this pairing of homologous chromosome, that is what called synapses. Yes, homologous chromosome. These two homologous chromosomes, they are similar in shape, size and structure. They are very similar in their shape, size and structure. Yes, and which are inherited from either parent. So each chromosome here, they are forming pair, that is homologous chromosome pairing. Yes, so why this pairing? Pairing क्यों होता है? So in between the chromosomes, here why pairing? To develop the attraction. Okay, and ये attraction की वजह से यहाँ पे एक synaptonymal complex form होता है. Now this will enter in the next stage, that is in Pachytene stage. So let's see the next stage, Pachytene stage. Okay, Pachytene. So what will happen here in this Pachytene stage? Crossing over takes place. Crossing over takes place. Pachytene. Okay. Crossing over takes place. So it is like the structure will be this is one chromosome. And here is the other one. So structure will be like this. Okay. So here we can say crossing over in homologous chromosome. Okay, here nucleolus disappearing. This is the nucleolus disappearing. न्यूक्लियोलस क्या हो रहा है? डिसअपियर हो रहा है. Pachytene, or we can say pachynemia, where crossing over takes place. Okay. Thick thread. Here the thread which is formed, that is thick thread is going to form. Thick thread. Chromosomes are like thick thread. So during this phase, the condensation of chromosomes that progresses and they become short and thick. यहाँ पे जो chromosomes हैं, chromosome become short and thick. Yes. And each of the homologous chromosome now show two chromatids so that यहाँ पे the bivalent appear. Okay, bivalent, and that composed of 4 chromatids, Pachytene. So this is what? Bivalent, or we can say tetrad formation. So number of chromosomes are 4. Number of chromosomes are 4. So each bivalent is known as tetrad. Okay, so in this phase here, if you'll see the number of chromosomes, chromatids are 4. We can say number of chromatids 4. 4 है, इसलिए each bivalent are known as tetrad. Okay, but each chromosome have 2 chromatid. One chromosome have 2 chromatids. So each chromosome are known as dyad. 2 dyad together makes tetrad. So here this is the Pachytene stage where we had seen here crossing over takes place. In crossing over, what will happen? Well, these chromosomes become short and thick. Yes, crossing over takes place. During the crossing over, the exchange of genetic material between non-sister chromatids. Here what will happen? Exchange of genetic material between non-sister chromatids. Of homologous chromosomes takes place. Yes. So this exchange of genetic material, that is only the crossing over. Yes. And this is very significant part of this phase. That result in genetic recombination. That results in genetic recombination of the parental character that leads to variations. So this point, the point where crossing over takes place, that is called as chiasmata. Okay, the point where crossing over takes place, that is called as chiasmata. And now After the Pachytene stage, it will enter where? In the Diplotene stage. Now let's move on. The fourth substage, that is Diplotene. Now in Diplotene stage, here Diplotene again comes from the two word diplonema. We can say diplos means two, nema means thread. Yes, Diplotene, or we can say diplonema. Two words. Diplo means what? Diplo, two. Nema, thread. Now in this phase, Diplotene phase, the homologous chromosome now start repelling each other, as we had seen in the earlier phase. Here crossing over. Yes. So in next phase, they start repel each other. So here homologous chromosomes now start repelling each other. Very important. As they will start repelling each other, now what will happen? They begin to separate. They begin to separate, and in this condition, the diagram will be like this. Let's say here in this phase, Diplotene, nuclear membrane, nucleolus will start disappear. Here the nuclear membrane and nuclear membrane disappear. Okay, start disappear. Here this nucleolus, it also start disappear. And the chromosomes will start repelling. जो क्रॉसिंग ओवर हुआ था वो अलग होने लगते हैं. So it will be like okay. So here diagram is like this somehow, and here you can say the point at which it takes place, that is what called as chiasmata. So this is what chiasma. Here this is the chromosome. Here you can see chromosome with two chromatids, that is sister chromatids, and these are with two chromatids. Diplotene stage. Okay, so here we can see the homologous chromosome, they start repelling each other, means they begin to separate. Okay, and at the point of crossing over, they remain attached, and thus here chiasmata are seen. Okay, and nuclear membrane and nucleolus, they start disappear. Okay, and they held only at chiasmata. So this is the Diplotene stage. And after Diplotene, here we had discussed homologous chromosome, they repel each other, means in the previous, if initially pairing and then this after the pairing crossing over takes place, and then here they start repelling, or we can say unpairing. We can say unpairing of homologous chromosome in Diplotene stage. Yes, because the synaptonymal complex start dissolving here, so unpairing, and this point is chiasmata. So chiasmata is what? It is actually recombination nodule. Here this chiasmata recombination nodule कब दिखाई देता है? जब ये unpairing होगा या desynapsis. Then at that time this chiasmata will be seen. Okay. So it is like something moved towards the periphery. Unzipping जैसा दिखाई देता है. So after this diplotene stage, where this chromosomes, they start repel each other. Now let's move on to the next phase of prophase 1, last phase, that is diakinesis. That is the prophase 1, fifth substage, diakinesis. Diakinesis. So during this phase, chromosomes continue to condense and shorten, and all the bivalent chromosomes separate and in contact at only one point. So here in diakinesis stage, all the bivalents chromosomes separate, and in contact at only one point. Diakinesis. Nuclear membrane disappear at this stage. Nucleolus also disappear. So let's see. This is our cell. Here nuclear membrane and nucleolus totally disappear, and spindle formation procedure will complete during this stage. The chromosomes, let's see. Here nuclear membrane and nucleolus completely disappear. Okay. Dotted means disappear. See the diagram carefully. Okay. So this is the diakinesis stage. Spindle formation is completed. This is the spindle. Ester formation. Yes. So during this phase, chromosomes, they continue to condense, and short chromosome continue to condense and shorten. and separation of homologous chromosome. Here separation of homologous chromosome. Yes, as separation of homologous chromosome, here you can see the chiasmata get shifted to the end of the chromatid. Chiasmata get shifted to the end of the chromatid. Chiasmata get shifted to the end of chromatids. And this, the process where chiasmata shifted, this is what called as terminalization. Okay, so this is the chiasmata shifted and this is called as terminalization. Yes, here in this phase, nuclear membrane nucleolus completely disappear. Nuclear membrane and nucleolus completely disappear. Okay. So this is all about the prophase 1, all 5 substages, and spindle formation procedure will also complete. Spindle formation procedure also complete. And dikinesis also complete. Okay, so we had studied about prophase 1, all 5 subphases. Let's see in short the summary. Yes, prophase 1. So first we had seen leptotene, coiling of chromatin, thread like chromatin. Yes, coiling of chromatin. Second, zygotene. What we had seen here, pairing of homologous chromosome. Homologous chromosomes. Yes, that is what? Synapses. Next is Pachytene, where crossing over takes place. Okay, crossing over takes place. Then we had seen Diplotene, where desynapses. Okay, and last one which we had seen, that is dikinesis, where nuclear membrane and nucleolus disappear. Yes, so here in this way we had seen the summary. So in short, you can easily remember these all phases. Yes, complete summary. So in this way we have completed with the prophase 1, and now we will continue with metaphase 1, anaphase 1 and telophase 1.